Hello friends, today I'll be explaining to you about the organization of DNA inside the human cells. Human cell is a microscopic structure and DNA, which is about 6 feet long, is present inside the nucleus of the cell. Can you imagine a 6 feet long structure fitted inside the human cell, which is a microscopic size? Actually, DNA is present in a very highly convoluted, compact form inside the nucleus. When I say it is present in a highly compact form, it seems to be very simple. But actually, the organization of DNA is a complex process. It involves lot many convoluted structures: a primary structure, secondary structure, tertiary structure, and a finally compact condensed structure, which forms a part of chromosome strand. But you don't need to worry, because with the help of our model, we'll be simplifying these things. And in this process, my colleagues Prathamesh Gatkal and Advait Shetty will be helping me out. The prerequisite for DNA organization is the histone protein. This histone protein is an octamer, and which is made up of 2 tetramers. Now, each tetramer, it consists of the following protein subunits: H2A protein subunit, H2B protein subunit, H3 subunit, H4 subunit. These 4 subunits, they come together to form a semi-histone, which then combines with the another tetramer to form an octamer called histone. Now, since it is very difficult to go ahead with this model, we will be taking the replica of histone, which is the smaller sphere, which will be acting as histone protein. Now, this blue color strand over here represents the DNA strand. As you can make out from the length, it is about 6 feet long. Now this strand will start coiling round the histone protein. It will give 2 coils on each histone protein. This coiling takes place throughout the strand of DNA. Now, as you can see, this structure is quite unstable and it can reopen at any moment. So, you require a stabilizing protein, which is an H1 histone protein. The protein which is being shown will be acting as a histone protein 1, which will try to stabilize the following structure. Now, this histone protein, along with 2 coils and a histone protein 1, is forming a 10 nanometer structure, which is called as nucleosome. And the strand in between the 2 nucleosome is called as the linker DNA. Due to the attachment of H1 histone protein, there is strain generated in the DNA strand, and this strain causes the nucleosome to come together, and such 3 histones come together to form a 30 nanometer structure. Now again, since it is very difficult to move ahead with the model, we have the replica of the structure. This another model will be explaining to you how DNA coils further. Now these 3 beads, Now the three beads, these three yellow beads, they are the replica of the histone proteins which had come together to form a 30 nanometer structure, and the blue color strand is the DNA strand which has coiled around the histone protein. Now such coiling takes place throughout the length of DNA, and such 30 nanometer structure is formed. Now, even though the volume of DNA has reduced to quite less volume, it is still not enough to enter into the cell. So, further coiling takes place. Now, the pink color strand which you are seeing is an imaginary strand, and this 30 nanometer structure starts coiling. This coiling further reduces the volume, but yeah, it's still not enough. As you can see, the length is still quite big, and since the structure is not condensed, it cannot enter into the nucleus of the cell. Next, to reduce the volume further, the supercoiling of the DNA takes place. This is the supercoiling of the DNA structure. This strand is also called as the chromatin fibers, which has formed a supercoils. As you can see, the volume is quite reduced, and still it is not further to move ahead with this model. We'll be taking another final model which will be forming a very condensed structure. Now this coiling, as you can see, is actually the supercoiled structure of DNA. Further, to reduce the volume, it will start coiling on an imaginary support. This wooden stick is an imaginary support over which the supercoiled structure of DNA starts coiling. Now you can imagine how compact the structure is. The coiling of the supercoiled structure reduces the volume drastically. But this is not the real phase. Actually, this coiling is very, very compact and very, very dense. As you can see in the video, the coiling tends to be very, very compact. And now, yeah, you can see how dense the structure has become. And this is just one single strand of the chromosome. This condensed form of the highly convoluted structure or the quaternary structure of DNA is just a small part of a chromosome strand. Such structures, they come together to form either the X chromosome or the Y chromosome of the DNA. And these chromosomes, these strands, they are held by a centromere, which helps to stabilize the final compact form of the DNA. And this is how, this is the final size which enters into the nucleus of the cell. Thank you.